So today we're going to talk about cellular respiration. Hopefully you have a little bit of an understanding of what cellular respiration is. You should be able to know what the reactants and the products are of cellular respiration. So if you just think about it, think about what do we take in. So we take in food, we take in glucose specifically, and we breathe in a certain type of gas. We breathe in oxygen. And we breathe out CO2 and just a little bit of water. So if you take your hand, breathe out, you can feel a little bit of moisture on your breath. So hopefully you can come up with the equation just by kind of thinking through what we need and what we breathe out. Hopefully that will help. But we're going to go into a little bit more detail today. This process is something that you need to be able to tell another person, so you should be able to go through this process and describe the different steps and what's going on in each step. So that's kind of the expectation as we go throughout this PowerPoint today. So first thing we're going to do is we're going to talk about some guiding questions. I'm going to have you stop and talk with a neighbor for just a second and see if you guys can come up with the answers to these questions. So question number 1, why do you need to breathe? And question number 2, what happens to the fat when you lose weight? Or where does it go? So I want you to write down the answers to these questions and kind of talk to a neighbor. See if you guys can come up with a good answer. I'll pause for just a second. You can pause the video if you need to. Okay, so we're going to come back to those questions at the very end. We'll see if going through this helps you answer those questions in more detail. So we're going to talk about cellular respiration here and kind of a more detailed definition. So go ahead and write this down. Cellular respiration is the set of the metabolic reactions and processes that take place in the cells of organisms to convert energy from nutrients into adenosine triphosphate, or ATP, and then release waste products. So essentially, we're kind of looking at our metabolism and kind of those different reactions going on and how we can take the energy from sugar or our nutrients and turn it into that chemical energy of ATP. So in this little diagram here, you can kind of see that same process. We're taking in oxygen, taking organic compounds, using the food and whatnot to create carbon dioxide and water. So where does this process occur? Well, it occurs in the mitochondria. So I'm going to have you guys draw this little diagram of this mitochondria. You guys need to know these different spots inside the mitochondria. So we're going to break it down even further to what we learned recently about this organelle, and also more than you guys have known in the past, that this is the powerhouse of the cell. We're going to go into a lot more detail. So you need to know a little bit more than just this is the powerhouse of the cell. So we've got the outer membrane and the inner membrane. These are called the cristae. They are like folds. So this inner membrane, to increase surface area, makes these folds all over the place. So we call those cristae. And then inside, we call this the matrix. So a lot of the process of cellular respiration will happen in the matrix. Some of it actually happens in this red space in between the outer and the inner membrane. We call this the intermembrane space. That's another one you should probably also add. So if you add another line here to show that that is the intermembrane space going on right there. Okay, so the equation for cellular respiration, we want to make sure we can write this down. You should know what molecule this is, hopefully. This is glucose. So we take any organic compound that we eat and we turn it into glucose. Our body just kind of does that conversion on its own. So if we need to have fat, like, it'll convert it into these sugars in order to be used in the mitochondria. So here's our sugar plus our oxygen, and then that creates carbon dioxide and water, and approximately, because this varies a little bit depending on where in the body we're at, about 38 ATP. So this equation is balanced. by adding these 6's in here, we can see that the number of carbons on this side are equal to the number of carbons on this side. The number of H's should equal the number of H's on this side, and O's should be the same on this side as well as on this side. Okay, so we're going to talk about some different stages of cellular respiration. The first one is called glycolysis. Glyco refers to sugar, and lysis means to cut. So we're going to take our glucose molecule and we are going to turn it into what's called pyruvate. Basically, we're going to take that sugar and we're going to split it into two. So we're going to take our 6-carbon glucose, and to split that in half, the pyruvate will end up having 3 carbons in each of the pyruvate. So we'll have 2 pyruvates to go into the mitochondria to get more energy. Now, this first process happens inside the cytosol. So go ahead and add this to your drawing here. This cytosol is not on that little drawing, so go ahead and add it. This is the cytoplasm. This is basically the liquidy part of the cell, not inside the mitochondria. So that's kind of important to note. The other thing that we see here is that these are electrons that are carried via NADH. I like to call this guy NAD. He's our buddy NAD, and he carries any of these electrons that we kind of get from this reaction into this third step called the ETC. We'll come back to this in just a second. So glycolysis is the first step. We take the sugar and we split it apart. When we take that sugar and split it apart, it actually creates some ATP. We call this substrate level phosphorylation. Phosphorylation because that phosphate is added, so we get that ATP. So we take ADP and we add a phosphorus to get the ATP. So we've created some energy here. Net, we get 2 ATP out of here. And then this next step goes into the Krebs cycle. The Krebs cycle takes that pyruvate, swishes it around. We get some electrons that are carried again. This time we have another friend. His name's FAD, FADH2, and he also carries some electrons over into this electron transport chain. We get some ATP from this step, and then we go over to the ETC. Sorry, that's my dog. She wants to go outside. The electron transport chain, I like to call this the ETC, kind of like the EAC, the Eastern Australian Current, from Finding Nemo. Dude, noggin. So the ETC, which we'll get to more detail on that in a minute. So all 3 of these steps create different amounts of ATP for our body. So this is why we call the mitochondria the powerhouse of the cell. Inside this membrane here, we can actually see some different proteins involved in this process inside of our membrane here, inside that mitochondria. So here's the matrix. So inside that yellow spot, this would be the inner membrane, and this would be just outside that. So this is basically a bunch of proteins that are involved in this process. The purple are the nuclear encoded, and the red is the mitochondria DNA encoded. Basically, the difference here is you can see that mitochondrial DNA, yeah, that's not fun. I'm not letting you go outside. You'll have to wait. The red here is inside the membrane, in that inner membrane, and that purple, that nuclear encoded, kind of spans or goes across the membrane. So that's the main difference there. It's funny because you guys always tell me that the mitochondria is the powerhouse of the cell. That's the only thing we remember from 7th grade, is that the mitochondria is the powerhouse of the cell. So yeah, this is where the mitochondria goes from being the powerhouse of the cell to the site of oxidative phosphorylation. Yeah, it's pretty fancy. All right, so this is an aerobic process, meaning it requires air. So going through into the mitochondria actually requires the presence of oxygen. Without oxygen, this process is actually called fermentation or anaerobic respiration. You may have heard of the word fermentation before. You might know what it refers to, and these pictures might help you. Anaerobic, ana means without. Aero means oxygen. So this is a process that happens without oxygen. The fermentation process basically happens when the Krebs cycle can't actually occur. So the only step that can actually happen is glycolysis. So in our diagram on this side, you can actually see that the glucose can be split and we get ATP from there. We get our 2-pyruvic acid from that step, but we actually can't go and do the next step. We are kind of stuck without oxygen. So in the Krebs cycle, we would be able to go and keep breaking that pyruvic acid down and get more ATP, but we're kind of stuck. So our bodies and different bacteria, fungi, mold will actually go through this process called fermentation. So they will actually take that pyruvic acid and they'll break it down. There's 2 options. So in our body will create what's called lactic acid. You may have heard of this before, especially if you are athletic and you have heard of that lactic acid buildup. But in our bodies, when we're not getting enough oxygen, our body will go into this mode or this process that will require less oxygen, but still use that sugar that our body creates. So it actually creates what's called that lactic acid. Basically gives us just a little bit of energy, but that lactic acid kind of is built up and stored in our muscles. And the reason it hurts is because it's acidic, and your body, when your muscles are being worked, they're kind of tearing. So that lactic acid actually kind of hurts and stings those little tears because of the acid concentration. The other product is going through this step on this side. We get ethanol. So ethanol is our alcohol. And this is why we commonly think of fermentation as the process that creates alcohol. So lactic acid is not alcohol in our body, but that ethanol process, if it goes through this side, does create some alcohol. You can go ahead and add this to your sheet as well on number 5. So you can see where the byproducts are. These products over here, ethanol is one of those byproducts. This is just the chemical formula, this is the name. And then carbon dioxide is also the other compound that's created. So make sure that you also write lactic acid is another one of the byproducts. So we've also maybe heard of fermentation in the food making process. So depending on which organism is going through fermentation, it will create a different fermentation effect. So for example, sauerkraut or vinegar or cheese, or if you guys have heard of kombucha, wine, alcohol, soy sauce, all those things are going through some type of fermentation. So for example, when you make cheese, a lot of the times it can be a bacteria that is going through fermentation, or sometimes it's a mold. And you guys have probably heard of that. Aged cheese gets better as it ages. Typically those are mold producing. Vinegar is actually created kind of similar to the process of wine. Wine is just a bacteria that's added where it ferments over time, whereas vinegar actually uses a different bacteria that causes it to go through acetic acid breakdown instead of that ethanol. So it creates that acetic acid, which is basically the main compound of vinegar, the stinky stuff. Okay, so let's go into a little bit more detail about each of these steps. So we're going to start with glycolysis. So glycolysis is that very first step, like I said, breaks down that sugar, that glucose, into 2-pyruvic acid. So you can see in our organic molecule here that there are the 6 compounds. Basically, if we split this guy in half, we get these 2 pyruvates here. The enzyme that breaks it down is called pyruvate kinase. We know it's an enzyme because it ends in ase there. And by doing this, at the end of the process, we get a net of 2 ATP that is produced. So hopefully you guys can add that to your little diagram there. You also will notice down here on the bottom we've got NAD. Our buddy NAD here is going to be our electron carrier that is going to carry electrons into that very last step. So it starts out as NAD with a plus sign and then adds that H. That's where it's kind of carrying those electrons. I would like you guys to also add where this happens for number 6. This process, like we said a minute ago, happens in the cytosol. So this is not actually happening within the mitochondria. Okay, the next step is the Krebs cycle. We also call this the citric acid cycle because we are creating that acid in here throughout the Krebs cycle as we kind of break some stuff down. So in this step actually happens twice because in the first step we created 2 pyruvic acid. So for every 1 glucose, we get 2 pyruvic acid. So everything in this step we are going to times by 2. So pyruvic acid is converted in this very first step into acetyl-CoA. So we can see that NAD kind of picks up an electron here. We're going to kick off one of those carbons and send it off as a carbon dioxide, and then this CoA molecule is going to come in and attach, and that's how we get that acetyl-CoA. The next step is it kind of goes in through this cycle. That CoA kind of gets kicked off. Then we kick off another 2 carbon dioxides. That's where those 2 kind of come from. And then we'll pick up some more electrons, create some ATP, and pick up a couple more electrons. So in this step, I am not going to require that you know all the details in the Krebs cycle because this cycle gets super intense. But you do need to know that there are 3 NADs created here, right here, times 2. So we have a total of 6 NADs that are created. 2 ATP are created because we have 1 down here, but we have done this process twice because there's 2 pyruvates. And there are 2 of these guys after we've done it twice. So there's 1 for each pyruvate, but we have 2. So our products from this reaction are 6 NADH, 2 ATP, and 2 FADH. So that's kind of what we're getting out of this process. And just make sure you guys write that down. And where is this happening? So you can kind of see over here in this corner, occurs in the matrix of the mitochondria. So this is happening inside the folds, inside that space right in the middle of the mitochondria. Okay, the last thing, oxidation of glucose. Basically, if we're going to oxidate glucose, our goal, or the product that we're actually getting out of this step, is carbon dioxide. So that is one of the products of cellular respiration is carbon dioxide. So this is the main step of where that carbon dioxide actually comes from. Just a little bit further with the citric acid or Krebs cycle. This, you can kind of see where this carboxyl group is. This is from our pyruvate. We actually remove this pyruvate or carboxyl acid from the if you remove a carboxyl group, it actually creates a carbon dioxide. You can kind of see carbon dioxide. So that's kind of where that carbon dioxide comes from. And then over here, we can see that NAD pick up that electron that's kind of created here. That's the oxidation step. And then this acetyl-CoA comes from this group right here. This is a new functional group that I don't think we've talked about. This is an acetyl group. And this is why we call it acetyl-CoA, because this group is attached to this bigger molecule here. I think we've answered everything for this step, so we're going to move forward. Okay, now the last step in the Krebs cycle is called that electron transport chain, the ETC, we are going to be inside the cristae. We are actually inside that membrane, in between the matrix and the inner membrane space. So here's our inner mitochondrial membrane right here. We've got a bunch of these proteins that are going to act as our kind of electron carriers. And you can kind of see NAD and FAD in here. This is where they come in and they do their job. So I like to think of these as like basically the turtles that are carrying the electrons to the electron transport chain. If you guys have seen Finding Nemo, the Eastern Australian current is like taking the turtles, they like get on the current and then they flow. This is basically the same process but with electrons. So I like to think of NAD and FAD as my turtles, and my electrons are kind of flowing through. And as they kind of pass along these series of enzymes, they actually come up with a bunch of energy. So this is the powerhouse part of the mitochondria. We actually call this process chemiosmosis, basically using osmosis here to create a bunch of energy. So we're going to be pushing some things across these proteins, kind of with their membrane flow. So we don't actually need energy for this process because everything is just kind of going with the flow, kind of with that, current theme. But these electrons are going to be pushed out into the intermembrane space. These H's are going to be pushed out. And this is actually what is going to drive the ATP synthase. And you're going to get about 36 to 38 ATP in this step alone. So that's quite a bit of ATP just by pushing electrons through these proteins and then pushing them back through at the very, very end here. So this, you've got where did it occur. Hopefully you wrote the inner membrane space of the cristae. You've wrote down the process that drives ATP synthase, which is chemoosmosis, and the net yield of ATP of 36 to 38 ATP. So to be a little bit more specific here, you can kind of see all those the hydrogen-free electrons that are coming in through here. The electrons are kind of pushed across this yellow section here. This little arrow is representing where the electrons are going. Those hydrogens are pushed up, and now we have like this imbalance of hydrogens. So we have a bunch of hydrogens out here now, and we're going to actually push them back through this protein over here. We call this one ATP synthase. You can kind of see in the little key over here that ATP synthase. So we've transported all those electrons, then we're going to kind of shoot everything through this protein here. I like to think of this like a generator because it actually like spins, like it's got this detail in it that causes it to spin, and basically kind of like water flowing out of like a dam. These electrons going back through to the other side is going to create that energy that we need to take that ADP into the ATP. So that's kind of that last step there. So why do we need oxygen then? The point of the oxygen is actually to kind of pick up those electrons. So the electrons will finally reunite with oxygen at the end of this process. So oxygen is going to be needed in this process to come pick up any of those leftover oxygens, or to go to the oxygen at the very end of this process. So without oxygen, this process just shuts down and goes through fermentation. So we need those oxygen molecules to come pick up the electrons. We can also use those oxygens and those H's to create some water. So in this step, oxygen will come in, and these hydrogens here will connect to it to create our other product of water. So you can kind of see this reaction here is super important to show kind of that other thing that's produced. So 2 H's, a couple of electrons are picked up because these are H pluses, and then our oxygen is connected there. All right, so just a quick little review. Hopefully you guys know the 3 steps. The first stage is glycolysis. The second step is the Krebs cycle. And the third cycle is the electron transport chain, also known as the oxidative phosphorylation. The reason we call it oxidative phosphorylation is because it requires oxygen, and that phosphorylation of the ATP is happening, so we're getting a bunch of ATPs created. All right. So take a look at this diagram, and I want you guys to fill out what is missing on number 11. So the ATPs there, the names of the cycles, where the electrons are carried, give you guys just a second to kind of fill that out. If you need to pause for a second longer, I'll let you pause. Make sure you guys note kind of the parts of the equation for cellular respiration that are present here. So our oxygen up here is a big key part of our equation. This goes in, as well as our glucose. So those are our two reactants, or the things that are going in to our process here. And then the things that come out, or the things that are produced, our products, that CO2 that comes from the Krebs cycle, and then our water that comes from the electron transport chain. So I want you guys to think for just a second. I want you guys to talk with your neighbors. How would you guys actually measure respiration rate? How could we measure how much someone is going through this process of cellular respiration? Pause this video for just a second. Talk to your neighbors. So we can see here that with the respiration rate in this little diagram here, basically they're trying to capture the amount of oxygen that is produced. So you can see that the fresh air, the air is allowed in, sorry, they're not capturing oxygen. They're letting the oxygen in into this tube. And then we're going to see kind of what is produced here. So in this case, most living things require oxygen, and the KOH will combine with the CO2 to form a precipitate. So at this step, this is kind of more intense. This stuff right here, this KOH, is actually going to create more precipitate as that CO2 is created. So we can kind of measure how much something has respired. Basically, other ways you can do this is basically just to capture the amount of carbon dioxide that is created or that's present. How would this rate differ between a cricket and a mouse? So I want you to talk to your neighbors for a second. How would the amount that we would capture from a cricket be different from the amount that we capture from a mouse? Go ahead and talk. You can pause this video if you need to. So between a cricket and a mouse, we actually can see a different level of respiration depending on its metabolic rate. So depending on how fast it's going to basically use that glucose. So you might think that a smaller organism may need less glucose, but that may or may not be true. It basically depends on its metabolism and how fast it actually metabolizes that glucose. So it may not necessarily be the size of the organism, but more or less its metabolic rate. So take a look at this graph for just a second. See if you guys can explain what's going on. Go ahead and pause if you need to. Okay, so on the left side, we always need to look at our axes when we're looking at graphs. And in this graph, we have the respiration rate, basically how much carbon dioxide is produced per minute based off of these different sugar molecules. So we can see that glucose obviously is the highest. Sucrose would then be the next, and then lactose is the lowest. You should hopefully remember that sucrose is a disaccharide. It's one of the most common disaccharides, and it is made of glucose and fructose. And lactose is also another disaccharide. It's got glucose and galactose. So if I were to kind of make an inference or kind of explain in some reasoning why I think that sucrose is higher than lactose, I'd probably say because it was easier to digest and break apart those sugars. And obviously that's kind of a educated guess. But we can see glucose obviously is the highest because it's a monosaccharide. It's just a single sugar. It can go straight into the process, where these need broken down first. So that's kind of how we would explain this. Here's another graph. This is talking specifically about the metabolic rates of these animals. And this is going to kind of connect what we talked about on one of our previous tests with the ectotherms and the endotherms. Remember, an ectotherm needs energy. It kind of moves to kind of warm itself up, whereas an endotherm is going to kind of keep that constant temperature at all times. So if the ambient temperature goes up, the outside temperature goes up, this guy is going to require more metabolic rate. His resting metabolic rate is kind of in the middle, where that temperature is kind of even. Whereas this guy, when it's really, really cold, is when we see that metabolic rate really high. So with this guy, an increased temperature gives that increased metabolic rate, and with this guy, a decreased temperature gives a metabolic rate. You guys hear my baby? She's making noises. All right, so hopefully at this point you guys can come up with what the purpose of cellular respiration actually is. Basically, the purpose is to turn sugar, or that chemical energy in sugar, into energy in the form of ATP, which is also chemical energy, to help power the cells. Where does this all happen? Now, we might say the mitochondria, but that is not fully true. Steps 2 and 3 happen in the mitochondria, but the first one happens in the cytoplasm just outside the mitochondria. And the third steps, what are the waste products of cellular respiration? Basically what is produced. So in these steps we have water and carbon dioxide. So I'm not going to answer these questions, but you should be able to answer these questions. So you guys can pause and answer these 2 questions with your partners based off of what you've learned. Hopefully you now have a different understanding of what these are. Make sure you write an updated understanding. Pause it if you need to have some more time to do that. Okay, I have 10 quick questions here that are just kind of some review type questions. Hopefully you guys can answer these. I'm going to go through them with you. If you want to pause and grab some whiteboards, you're welcome to, or you guys can just kind of go through this and kind of see if you can get the answers. You can pause in between each question if you need to. The first question, in order to produce energy, cells start with glycolysis. If oxygen is not present after glycolysis, what process occurs next? So if there's no oxygen, is it going to be electron transport chain, Krebs cycle, or fermentation? The answer is fermentation. So if oxygen is present after glycolysis, what occurs next? What process occurs next? Is it the electron transport chain, the Krebs cycle, or fermentation? This would be the Krebs cycle. A process that does not require oxygen is also known as what? Aerobic or anaerobic? This one should be anaerobic. In glycolysis, glucose is broken down into 2 molecules of, should be pyruvate. Where does the Krebs cycle occur? So the Krebs cycle occurs in the mitochondrial matrix. What gas is a waste gas produced in the Krebs cycle? Should be carbon dioxide. What enzyme is used in the electron transport chain to create ATP? Is this citric acid, pyruvate, or ATPase? Hopefully you guys can do this without even knowing anything about it. We know the enzymes end in ase, so it's probably ATPase, or ATP synthase is the other name for it. 8. Where does glycolysis occur? Cytoplasm, mitochondria, or the chloroplast? Hopefully we don't put chloroplast because cellular respiration is not done in necessarily in plants. Here, the chloroplast is a different process known as photosynthesis, right? Sorry, I need to go back. Cellular respiration actually does happen in plants. Cells that have mitochondria are going to go through cellular respiration. So basically all cells need energy, and so the mitochondria, if it's found, is going to be where this process occurs. But glycolysis itself, glycolysis actually happens in the cytoplasm, so just outside the mitochondria. So number 9, what process produces the largest amount of ATP? Fermentation, the Krebs cycle, or the ETC? It's going to be the ETC. And the last question. The oxygen required by cellular respiration is reduced and becomes part of which molecule? ATP, carbon dioxide, or water? And the answer is water. You guys hear those cute little baby sounds? She's so cute. All right, well, if you guys have any questions, you can reach out to me. Hopefully this was helpful. And good luck.